Hello, everyone, and welcome to lectures 3 and 4 for this week. Let's go ahead and get into it. Now, in lecture 3, we're going to be discussing the role of chemical senses in motivated behavior. Now, chemical signals or chemical signals play a central role in motivated and emotional behavior. They can be used to identify group members, mark territories, identify favorite and forbidden foods, and form associations among odors, tastes, and emotional events. Odor and taste also play a fundamental role in emotion and motivated behavior, such as chemical disgust and the more social forms of social disgust and moral disgust, or a hygiene disgust. Now, the chemical disgust, social disgust, and other cognitive appraisals related to disgust involve overlapping neural representations in brain structures, including the insular cortex, and these sensations of disgust to taste and smells are evolved into sensations related to morals, which is interesting to note as well. Now, olfaction, that's an interesting one in terms of human psychology because humans, they actually lack a vocabulary for olfactory perceptions and differences despite discrimination among thousands of odors. Now, olfaction is designed to discriminate if information is safe or familiar or to identify a signal from a receptive mate. So the most common example and the one that I use the most, I think, consciously in my day-to-day life is the smell that's coming from this food, is it going to be edible? So I pull out a bag of something from my fridge and it's been in there for a little bit. I can't remember exactly when I got the spinach. And so I open up the bag and take a whiff. And sometimes it smells like good old spinach, and sometimes it does not. The spinach is turned, and I immediately go ahead and toss that. And notably, that's providing us this really helpful information that if the smell is bad, that's probably telling us that the spinach has turned and would end up making me feel not great if I decided to consume it. There's also the smell thing come from a friend, or is it a smell of a stranger as well? I think those smells for people are often a little bit less conscious, but not always. Sometimes there are people or these familiar scents that we associate with a person that will bring us to that place or think of them. The receptors for smell and smell itself, sense, it interacts with the chemical receptors, which are constantly being replaced. So the olfactory receptor neuron lifetime, it's about 60 days. And the epithelial receptor surface varies widely across species. In humans, this area is estimated to range from 2 to 4 square centimeters, whereas in dogs, it's approximately 18 square centimeters. And in cats, it's about 21 square centimeters, interestingly enough. And in any given odorant, it stimulates a unique pattern of receptors, and the summed activity or pattern of activity then produces our perception of a particular odor. The olfactory system is estimated to contain about 400 kinds of receptors, compared with just 4 types, the rods plus the cones that we know about from the visual system. Some researchers, they've proposed that humans discriminate against as many as 1 trillion smells. At present, the actual number is unknown, but it's likely to be a lot more than is commonly believed. Now, the olfactory epithelium is the receptor surface for olfaction, and each olfactory receptor cell sends a process ending in 10 to 20 cilia to the mucosous layer, the olfactory mucosa. Chemicals then dissolve in the mucosa to interact with the cilia, and metabotropic activation of a specific G protein then leads to an opening of sodium channels and a change in membrane potential. Here we have an image of the olfactory epithelium. The air-carrying odorant molecules enters the nasal pathway and makes contact with the epithelium, where the molecules dissolve into the mucosa. if an olfactory chemical signal affects the receptors within the mucosa, proteins within the receptor are activated, opening sodium channels and sending signals through the uh, glomerulus glomerulua and mitral cells in the olfactory bulb and onto the forebrain. Sorry, I cannot pronounce that word to save my life. The glomeruli. Glomeruli. I think that's what it is. Glomeruli. Now, with the olfactory pathways, the olfactory receptor cells project to the olfactory bulb. Many olfactory targets, such as the amygdala and piriform cortex, have no connection through the thalamus. The thalamic connection projects to the orbitofrontal cortex, or the OFC, and that's used for the emotional, social, and eating behaviors. There's also these accessory olfactory system that utilizes some of those pheromones. So the pheromones are biochemicals that are released by one animal that acts as a chemical signal to affect the physiology or behavior of another animal. It's detected by a special olfactory receptor system known as the vomeronasal organ. There we go, connected to the amygdala and the hypothalamus. So in this figure that we have up above here on the left, there's a cat and it sniffs a urine-soaked cotton ball. In the center, we see that the cat raises its upper lip to close off its nasal passage. And then in the right, it follows with full gape, which is a response characteristic of the phleeman behavior that's mediated by that accessory olfactory system. Now, in the vomeronasal organ likely participates in analyzing pheromones, such as those in urine or sweat. And you may have seen horses or cats engage in a behavior that's known as the phleeman. When exposed to the novel urine from a cat or a human, there is this type of response where, as we saw here, the cat raises the upper lip to close off its nasal passages and to suck in air into the mouth. The air then flows through the duct on the roof of the mouth en route to the vomeronasal organ. Now, in human olfactory processing, humans have an acute sensitivity to behaviorally relevant smells. So humans can identify their own odor, the odor of kin versus others, and the odor of friends versus strangers with accuracy well above chance, interestingly enough. So if you were asked randomly, do you think you could identify your friends based on the smell of maybe a shirt or something? You may not expect that you'd be able to do it, but it sounds like humans have been found to do that above chance levels, which is pretty interesting. The body odors activate brain regions that are involved in a lot of the emotional processing. And the stranger odor activates the amygdala and insular cortex. I also think that's important to think about because there are many people who a particular smell or scent, it really brings them back to a particularly emotional memory. And I think that's really unique to some of the olfactory systems associations with memory as well, which is, again, I think a cool connection to try to make. And like we talked about before, the processing of body odors, it's mostly an unconscious and an automatic process. So it's not something where you're like, oh yeah, I know Daryl. Daryl has this smell. We're not able to articulate that very clearly. It's just not a conscious awareness process that tends to take place. It's just that researchers have found that if you are placing then Daryl's shirt in front of you, along with various others, you would more likely than not be able to recognize, oh, yeah, this shirt, if I were to guess, is the one that's Daryl's, and you'd be correct. Now let's talk about gustation. In humans, there are clear differences in taste thresholds and preferences that are apparent. There are what we call super tasters versus non-tasters. This is related to genetic differences in the ability to detect a specific bitter chemical, which is 6N propionuracil, or a PROP. which I would prefer to call it. And the bitterness is then there's associated with this allelic variation in the taste receptor gene. The specific one is TAS2R38. And people who can detect these minute qualities of that prop find the taste extremely bitter, whereas those who do not taste prop are who don't like taste the prop as being bitter are then the non-tasters. It's also interesting to note that children are much more responsive to taste than adults. So by age 20, humans have lost at least an estimated 50% of their taste receptors, which is interesting to think about. And I think it's interesting to think about that too. I do a lot of work with older adults, and sometimes when you ask them about food and how food tastes, they'll say, "Oh, everything seems like it's bland. It doesn't taste like it used to." And again, that's just part of the typical aging process. Each of the 5 taste receptor types respond to a different chemical component of food. So those are the sweet, sour, salty, and bitter. And then there is this 5th that is called the umami receptor. I tend to think of the umami flavor as being almost like the savory type taste. And it's especially sensitive to glutamate. And one of the most clear connections with the umami is like MSG. MSG is usually used to be the kind of trademark flavor of umami, I think. The taste receptors, they're grouped into taste buds, each containing several receptor types, and the gustatory stimuli interact with the receptor tips or the microvilli. The ion channels open, leading to changes in the membrane potential. Cranial nerves, 7, 9, and 10, form the main gustatory nerve. The solitary tract, gustatory region, and the insula is dedicated to taste, and the primary somatosensory region is responsive to the tactile information, such as localizing taste and textures on the tongue. The gustatory nerve to orbital cortex, the mixture of olfactory and gustatory input gives rise to the perception of flavor. Now, areas in the right orbital cortex mediates the pleasantness of tastes, whereas the same region in the left hemisphere mediates the unpleasantness of tastes. And the gustatory projections to the amygdala and the hypothalamus, they play a role in the pleasantness and strength of flavors. As far as impairments in the chemical senses and behaviors, impairments in the chemical senses can be profound, often subtle impacts, such as increased incidence of depression, decreased sexual satisfaction, deficits in personal hygiene, detriment in health amongst the elderly, failure to activate the disgust system, which is really important. Again, we're thinking about the food safety side of things. And the loss of olfactory and gustatory functions is referred as anosmia and hyageusia. These were notably early and prominent symptoms of the COVID-19 infection, and they're also early biomarkers of both Parkinson's disease and dementia, often preceding the emergence of motor or cognitive systems. So it's, again, I think, interesting to think about the sensitivity of those specific pathways. All right, welcome now to lecture 4 of this chapter. This lecture is going to be talking about the control of regulatory behavior, such as controlling eating. Now, eating disorders lead to being either overweight or underweight, and another key term that we want to be familiar with is going to be obesity. This is the excessive accumulation of body fat, which of course would be in the overweight category. On the other end of the spectrum, in disordered eating, there would be anorexia nervosa. This is an exaggerated concern with being overweight that leads to inadequate food intake and often excessive exercising. And it can be either or. Regardless, it can lead to severe weight loss and even starvation. And importantly, we want to note this part. It's not it's not just like dieting, right? It's not just not taking in much food or cutting calories. It's taking this to a further extent. It's really this individual is seeing themselves as being an overweight individual, even though objectively, as we look at the person's body weight, body fat percentage, things along those lines, it is in either healthy ranges or below healthy ranges. But that is just not the way that this individual sees themselves in the case of anorexia nervosa. Now, with the digestive system controlling eating, there is the enteric nervous system, which controls digestion. There's the detector cells that keep track of the level of each nutrient in the bloodstream. And three nutrients are extracted. Each is a specialized form of energy reserve. So there's the lipids, which are fats, amino acids, which are the building blocks of proteins, and there is glucose or sugar. The body needs to be appraised of how well the breakdown of food is proceeding, and a feedback mechanism then provides such information. Now, notably, as we're going back to those three nutrient types, glucose is the body's primary fuel source, virtually the only source of energy for the brain. That's because the brain requires glucose even when the digestive tract is empty. The liver, in that case, acts as a short-term glycogen reserve. So when the blood sugar level falls, the detector cells tell the liver to then convert glycogen into glucose for the release into the bloodstream. When food then reaches the intestine, it interacts with receptors to trigger the release of at least 10 peptide hormones. So I think one takeaway that we want to have there as we're thinking about the importance of glucose in the brain is that although there is this backup system that exists, make sure that we are feeding our bodies, right? I think something that I've noticed in people in my life is this increasing move towards not eating in the morning, kind of skipping breakfast and going on along the day. Now, that's putting your body into a state where it hasn't eaten for an extended period of time, over a long period of time, and it's the body needs and the brain needs some of those fuel sources in order to continue functioning well. So I think that is just an important consideration to try to be making. Here we have a nice image breaking down the digestive system a bit more. So the digestive system functions mainly to break down food, and the body needs to be apprised for how this breakdown is proceeding. There are feedback mechanisms which then provide such information. So when food reaches the intestine, it interacts with receptors in the ENF to trigger the release of at least 10 different peptide hormones, including uh, choline, uh, stokinine, or CCK is how I'm going to prefer refer to it, or glucagon like peptide 1 or GLP-1 and peptide YY, PYY. By its release, as food is absorbed, each peptide hormone acts as a satiation signal that inhibits food intake. So, for example, when the CCK is infused into an animal's hypothalamus, the animal's appetite is then diminished because that is one of those triggers that's essentially telling the brain that, oh, I'm full, I'm not needing more food at this point. Now, we're thinking about a couple of different problems that can take place with eating. There can be the aphasia. This is a failure to eat. It may be due to unwillingness to eat or to motor difficulties, especially when swallowing. Following, they can occur rather following lesions to the lateral hypothalamus. And researchers have found that electrical stimulation can actually then elicit feeding in these cases. On the other end, there can be hyperphagia. This is a disorder in which an animal overeats, leading to significant weight gain. This can be observed when there have been a lesion made to the ventromedial hypothalamus. And in this case, an electrical stimulation will then inhibit feeding if that is then placed. And again, I think it's when we're thinking about these, it's almost like the gas and brakes in a car, right? So here, it's almost as though there's this lack of gas for you to go and start eating, wanting to eat, have this desire to do that. Um, or it could also then just be you may want to just having difficulties actually being able to get the food in. Whereas the hyperphagia, there's difficulties with being able to apply the brakes. So we're starting to eat, but then we're eating and eating and eating, and the body isn't saying, okay, we're full, we're fully satiated, we can stop eating at this point. Damage to the lateral and ventral medial hypothalamus and the paraventricular nucleus has multiple effects. Leads to changes in hormone levels, especially insulin, as well as sensory reactivity. So changes in taste and attractiveness of food, the glucose and lipid levels in the blood, and the metabolic rate. So importantly, the hypothalamus receives inputs from the enteric nervous system. That's given the brain information and the hypothalamus information about the blood glucose levels. The hormone system also is providing input to the hypothalamus, like the information about the level of appetite diminishing CCK and the parts of the brain that process cognitive factors. The cognitive control of eating, the pleasure and its absence are really these cognitive factors in controlling eating. So in this case, there's the amygdala, which projects the hypothalamus that alters food preferences and it abolishes taste aversion learning when it's damaged. Uh, there's also then the orbital frontal prefrontal cortex, or the orbital prefrontal cortex, that receives input from the olfactory bulb, and damage there may decrease eating because of diminished sensory responses to food odor and perhaps taste. As we're thinking about this clinically in regards to diets and rhythms, so the effectiveness of different diets indicate that people generally lose weight in the first months or year. But by and large, these individuals usually gain that weight back. And in many cases, their weight actually increases over the baseline before they started the diet. The challenge is that there's little that's known about the cognitive regulation of eating related behaviors or how elements related to control of eating work together. But there has been some recent research by Dr. Karis Katavicio and colleagues, which found that the optogenetic stimulation could either drive or reduce feed-seeking behavior through the effects on neurons in the lateral hypothalamus. This work demonstrates a mechanism whereby cognitive activity can control eating. Then if we're thinking about the control of regulatory behavior in regards to drinking behaviors, it's important to note there are these two different types of thirsts that exist. So one type is the osmotic thirst. This results from an increased concentration of chemicals known as solutes in body fluids. By drinking water, the body's able to then restore the solute concentrations. There's also the hypovolemic thirst. This results from a loss of overall fluid volume from the body. And so individuals then drink fluids other than water to restore the nutrients. Now, again, as we're just thinking about the human body, it's made of about 70% water. And dissolved in that water are chemicals that participate in hundreds of reactions that are necessary for bodily functions. So essential homeostatic mechanisms really are used to then control water levels and therefore the chemical concentrations within this pretty narrow limit. The concentration of participating chemicals partly determines the rate of a chemical reaction. So as we talk more about this osmotic thirst, that deviates from the ideal solute concentration to activate systems to then reestablish it to that ideal solute concentration in the body. There's this message that's sent to various hypothalamic areas to stimulate us to drink, which is then shared. And it motivates us to get up and drink a glass of water. Now, there also can be this water intoxication. We were talking in one of the previous lectures about how we're needing to fit in some of these tight zones for some behaviors. So water intoxication, that's when the body tissues swell due to excessive fluid. This essentially ends up drowning the cells in fresh water, more or less. So it's drowning yourself at the cellular level. The water intoxication develops after sweating heavily by running a marathon in hot weather, and then drinking too much water without electrolytes to actually be able to be absorbed properly and used in the body. On the other end, there's the hypovolemic thirst. This thirst arises when the total volume of the body fluids decline, motivating us to drink more. This motivates us to drink flavored beverages that contain salts and other nutrients. The kidneys end up sending a hormone signal, angiotensin, that then stimulates the midline hypothalamic neurons when the fluid volume drops. Now, that is the end of lecture 4, and I will see y'all soon for lecture 5.